Hello, welcome to the Open Wound Research Podcast. And I'm very excited today to welcome our newest guest. Some would call him a victim, but I call him a guest, uh, Mitch Sanders, or as some know him, Dr. Mitch Sanders. How are you doing, Mitch?
Very well. Thank you. Thank you for having me.
It's my pleasure. It's my pleasure. We have met a couple of times in passing, and I appreciate you agreeing to come on the show. I see a lot of you on LinkedIn, and I've talked to a lot of people about you, and you just have a very fascinating background in wound care. And I'm just so excited to speak to you for the next hour.
It'll be fun. Look forward to it.
Great. Well, as I usually do, we're going to do a quick introduction so that we can tell the story of Dr. Sanders a little bit, or Mitch. Mitch is a wound scientist or a biologist, depending on what day of the week it is. I'm not sure which one you prefer. We'll get into that. He holds a PhD in wound biology from the Worcester Polytechnic Institute. He then went to MIT where he did two postdocs. One postdoc was not good enough. He did two, one in protein biochemistry and the other in cell biology, I believe, pathogen cell biology. At which point he became a serial scientist, entrepreneur, and C-level executive. He has, by his count and my confirmed count, 65 peer reviewed papers and multiple worldwide patents. He's currently a chair of the communications at the Wound Healing Society. He's also currently the chief scientific officer of Pro Dev Labs, an R&D and preclinical clinical research organization. His lab focuses primarily on preclinical and translational research in chronic inflammatory disease, including wound care, infection, biofilms, cancer, and traumatic brain injury. So, with that mouthful said, Mitch, welcome again. And you've got a great resume here and a lot of work that you've done in the community.
Thank you. For the Wound Healing Society, I've been involved for, it seems like 15, 20 years. I'm no longer chairman of the communications committee. I just help out. I think the new chairman is Lindsay Pullen, who works for me, was the past chairman. I think Jordan Yarin's now the current chair of that committee.
Perfect. Well, correction received. So, where we get started with every podcast is what got you into wound care? So, you finish up at MIT, uh, you're doing all this fascinating work. And how did you end up in wound care? What's the story there?
It's a very fun story. So when I left MIT, I worked for a company for a year. I always wanted to start my own company, but I was afraid I would lose my job because you kind of get lost in a big company. And after a year working for a firm, I realized that they weren't going to be around very long, and they were getting ready to close their doors. And so I set up my first company. It was called ECI Biotech, and the first patents that I wrote, we developed a diagnostic technology for a foodborne pathogen, Listeria monocytogenes, which isn't just for breakfast anymore. It causes foodborne illness, and if you're pregnant, you could even lose your child, have an early spontaneous birth. Um, and I developed a sensor for a protease called MPL, a diagnostic sensor that would turn blue or a little frowning face on your hot dog package if your food was spoiled. And I raised $1 million from a guy at MIT. And then I sat down with, developed the technology with a team of about 4 or 5 kids from WPI. And then I sat down with all the meat processors in the country, Pilgrim's Pride, Purdue, Keystone, all the big names. And they said, Mitch, this is incredible. I want to hire for you head of R&D. I said, no, I don't want a job. I want to bring this, sensor technology so that people don't get foodborne illness. And they said, well, your business model is different from ours. I said, what do you mean? They said, well, our business model is that somebody eventually gets sick and they die, and then their family can sue us. Your business model is mom and dad take fresh chicken home and know that it has Listeria or Salmonella, and they sue us today. And I said, oh crap, I spent $1 million developing a technology that nobody wants. What am I going to do? And and I looked down at my hands and I said, well, I could make a Band-Aid change color if you have an infection. So I went to Johnson Johnson Ethicon. They gave me 4.5 million to develop the technology, a broad spectrum diagnostic for chronic wound infection that covers all the major pathogens, both fungal and bacteria. Doesn't cross-react with commensal bacteria. Doesn't cross-react with host proteases, MMPs, 2,9, human neutrophil elastase, and we developed both a rapid point of care, a lateral flow diagnostic, as well as a band-aid that turned blue if infections do. And then, unfortunately, when we got nearly finished with the prototypes, J&J sold off Advanced Wound Care. It became Cystogenics. So we got a $2 million grant while they were kind of getting reorganized, re-signed with Cystogenics, and got the products pretty much, had enough clinical data to support that they were really going to work. And then unfortunately, Cystogenics got acquired by KCI. and and then so the product was kind of stuck in private equity for about a year, and then it came out as a little company called Wound Check, and our product's called Wound Check Bacterial Status, and that's the rapid point of care, the one that tells you in 5 minutes whether you have a wealth of proteases in the wound that suggests your wound's not going to heal. And it's a good, it's a 510K cleared. I think it was a de novo clearance. Dr. Serena, Tom did a ton of clinical studies on this product showing that if this sensor goes off and it goes off again, your wound's not going to heal. And so instead of doing the classical sensitivity and specificity and area under the curve for a diagnostic, we had to go, and because there was only one primary endpoint for wound care, which is healing, we had to show that if the diagnostic goes off, your wound's not going to heal. So instead of doing a simple study with a couple hundred patients, this turned out to be an enormous study with like 650 patients. And finally, the product came out, and but it took quite a while. It was quite a journey. Our first company we had up and running for about 16 years, and we developed diagnostics for other fields of use like periprosthetic joint infection and oral care for J&J Consumer. We had money from Strike Orthopedics for periprosthetic PJI, periprosthetic joint infection, and we really had a blast developing diagnostics. And after the company was kind of winding down, the products were coming out, I joined a consulting firm, investment bank, and then realized I still wanted a lab. So I went from, I literally had no business card. And in the first couple of months, I brought in a couple of million into the company to develop new products. And I was like, well, I guess I need a lab. So I built a lab in the middle of a consulting firm. And that was Alere Health. Worked for them for about 9 years, and then more recently used my equity to buy my lab out and start all over again and build a new CRO that's just focused on wound care for R&D, preclinical, regulatory, some strategy, a lot of IP portfolio management, and what have you. This has been a real blast. So over the past, I don't know, 15, 20 years, I brought in 65 million to my lab to develop wound care products, many of which you don't know that I worked on, because oftentimes we never get to talk about it. But you'll see the patents that come out or what have you. And over the course of this past year, we've had about 30 projects that we've been working on, both in R&D and preclinical and strategy, and helping clients get through the FDA hurdles and things like that. So it's been really fun.
And that's quite a winding journey. So let me just recast that real quick. So you went from chicken to
I went from,
wounds.
yeah, yeah.
So from chicken
You know,
to wounds to private equity and back again.
yeah. Well, but even in the consulting firm investment bank, I was an oddity. I was a scientist, and I didn't know what I wanted to be when I grew up. And I was like, I could be a. Do I want to be a venture capitalist? And I was looking at some job offerings there. Um, do I love running companies? My favorite thing was doing, business development is what I'm really good at, and cutting deals and bringing in business development funding to develop new products. And so, even when I joined Alere Health, I already had two startups in addition to kind of winding down ECI, and I sold those startups to Alere Health, and that ended up being a real benefit. We were making dressings that were the company was called Firefly, but the brand was called Pure Mend. We were making dressings that had both lemongrass and Chinese cinnamon in them, and they're tremendously antimicrobial, but they also smell amazing. So we're going to make chronic wounds smell nice for the first time ever. Um, and then we also worked on debridement. The problem in debridement, as you know, is that there's no good in vitro model for debridement. And so when we were looking at Santal as the gold standard, we were saying we can't develop a new drug for debridement until we develop a good in vitro model. And so I convinced a company in the Midwest, Hollister, to give me $1.5 million to develop a model and develop a drug. And the funny thing was, first thing I said to him is, now I need $500,000, and the first thing I'm going to do is I'm going to buy a smoker. I'm going to cook brisket. I'm going to cut off the burn ends. I'm going to chop them up in little squares and see if I can dissolve burn ends. And if I do, then we have a debridement model. And it turned out it really worked. And then we could do a high throughput screen for chemicals and come up with a non enzymatic debriding agent that's almost like a meat tenderizer for wounds. But based on clinical testing with about 12 patients, we can debride a wound in less than 10 days with a product that really is a meat tenderizer. It doesn't have any enzyme at all. And it's really cool, and it's inexpensive. And when we were initially working for Hollister on the project, they wanted a drug, so we convinced the FDA it was a drug. When we realized that they were getting out of advanced wound care, we had to take it back and then try and convince the FDA that it was a device, and they said no, we still think it's a drug. But we're going to get it launched in Europe and then hopefully find a commercial partner to sell the product.
That's uh, that's some fascinating work. I think if we can take a step back, you've been in wound care research for 15 plus years thereabout. Um,
Yeah,
how
almost almost
how is the
20 years.
Oh wow, okay, 24. So how over that time have you seen wound care change? You're sitting kind of at the fount of innovation, so to speak. What you've seen different demands over time. So compared to, let's say, the early 2000s to the mid 2000s to current, how has the demand for different research innovation changed from where you sit?
And I'm coming at it from I do a lot of cell tissue product work as well. So we do a lot of placental derived tissue and work in that whole camp cellular, acellular matrix like product space. What I can say is, you know, when I started in this field way back in 98 and 2000, getting into wound care, we were just at the foundation of thinking about infection and thinking about biofilms. So before we used to think about Phil Bowler's old premise that you have critical colonization. So if you once you have critical colonization, you're going to have an infection. I think what he was truly describing is a biofilm. And then with all the great work that Greg Schultz has done and some of the work that we've done, we've shown that biofilms are really persistent. They're resistant to mechanical shear. They're hard to treat with antibiotics because they're polymicrobial as well. And so that whole field has blossomed. And so there's a lot of new innovation products that have come out over the years that are starting to address these new fields that how to address biofilms. Now, the sad thing is, or the most difficult thing is, it's the FDA still doesn't recognize biofilms, but we're getting to a point where we're rewriting the guidance documents from 2006 with a whole group from the Wound Care Collaborative Committee, and I'm chairing this with Wendy Cole, and we're rewriting um, the entire subcutaneous, wound healing guidance documents from the FDA. And I think there we're going to start to bring in biofilms. We're going to start to bring in all these new areas of research that are so important to understanding how to diagnose and treat a wound, right? And so the FDA probably will push back on a bunch of these, but hopefully we'll get some through so that this guidance document can also be a little bit forward thinking to the new products that are coming out. And then think back in 2000, uh, back then we thought of something novel being just a collagen dressing. Now we have all these advanced therapies coming out that really um, not just heal wounds, but also probably potentially prevent recurrence. So, I my feeling is there's a lot of medical device products that can cover a wound, but do they truly heal a wound? I think the answer is no. If you pull the wound out and let's say you heal a wound in a diabetic mouse and you cut the wound out, cut the tissue out that healed, and you pull it apart, you don't get the tissue integrity strength that you would with unwounded tissue. There are some new products coming out that you actually do. And so, where I see us going is we're starting to get to the bottom of the underlying condition of the wound and understand the mechanism of action. And that's what our ProDev Labs is really focused on, is helping people understand how their product actually is better from product X or Y, and uh, what are the gene signals, what genes are turned off and on to cause reepithelialization or macrophage plasticity or quell the inflammatory phase. So I think we're getting down to the really understanding the mechanism of how some of these important drugs, devices, and biologics work. And what we'd love to do with the FDA is hopefully try and harmonize clinical guidance so that we can design trials so that when industry goes to design a trial, they're doing it in the best way possible, as well as how they design their preclinical studies to better understand the modalities of what's going on in the clinic. And the problem with these delayed healing models or even acute models is that none of them are perfect, and they're not exactly just like a human. The pig skin is closer to human skin, but the a wound is so acute, it heals so quickly that it doesn't tell you much. And then in the diabetic mouse model, the problem is that it's not truly chronic. It's a delayed healing model. It can tell you some things, but you may still miss some important things that you may be better off also doing some ex vivo models with human skin to better understand whether your drug or device is really going to work.
A question about models. I mean, as a stat guy, as a modeler, I really think in terms of models. So, in terms of the types of, let's talk about chronic wounds for a second. Dr. Yu, a couple of podcasts back, made a comment that really stuck with me in that the models that are being used to test products are on acute wounds. The majority of human wounds that we see are chronic wounds. And those are two similar but different universes. What When you start looking in terms of chronic wounds, how do you as a scientist attempt to approximate what's going on there? You've talked a little bit about it, but could you kind of drill down in how you approximate chronic wounds?
Right. So I really like the diabetic mouse model, the delayed healing model, because it has a pretty good inflammatory phase. And if you can quell the wound is red hot for 4 or 5 days. It gets bigger, it enlarges by 20%. And if you can quell that inflammatory phase, you're probably going to be able to quell the inflammatory phase in a chronic wound. From a proliferation point of view, the diabetic mouse is great because you have both early, mid, and late proliferation of the tissue, and you can see profound differences in gene expression for keratinocytes and fibroblasts and macrophages, and better understand who the important players are and who's directing the healing. That model is pretty good for angiogenesis by day 17. These chronic wounds, because we do 10 millimeter punches, they're pretty big when they start, and they take 24 days to really heal. And if you have a product that is profoundly different, you may, an acute wound in a mouse would heal in 14 days. So if you can speed up the time from 24 days to 14 days, you probably have something special that's going to work in a chronic wound. And what we see is a lot of these products, some of them don't do anything, and we're like not really impressed. Others quell a phase or are great for the inflammatory, quelling the inflammatory phase, or great for proliferation, or great for reepithelialization. The other cool thing about this model is you can stratify products. You can go in and say, well, first I need something to quell the inflammatory phase and mop up the proteases. Then I want to work on proliferation, macrophage plasticity to direct, migration and then reepithelialization. So because the model takes some time, you can also add redox inhibitors to delay it even further. You can study the wound for some time, right? The challenge with these mice is they're diabetic, they're profoundly diabetic. They're so fat, they're under so much tension that, most when you think about a rodent model, the skin's really loose and usually can just kind of contract and doesn't re-epithelialize well. In these fat diabetic male mice, the wound is under so much tension that they can't just purse string close. They have to they have to migrate to close, and so that's the benefit of the model. But it's, as I say, it's not perfect. But usually, what we see by gene expression and what we used to do, many years ago, we used to do qPCR. The problem with qPCR is there's a lot of pipetting, so there's potential for pipetting error. There's also the potential for amplification bias, and because it's relatively expansive, you only do 30 to 50 genes, and you're like, well, that's enough. And you always pick your favorite genes, and you may be missing the whole biochemical pathway, right? So what we do now in our lab is we do nanostring, and we have a custom code set with over 300 probes, covers all the phases of wound healing, macrophage plasticity, scarring, fibrosis, angiogenesis, and we can to a T get p-values that are strongly significant. And in addition to the mouse code set, we have a human code set as well, so that we can do when people are doing these pilot clinical trials to really understand their mechanism, we can go in there and really show that what we're seeing in the mouse is exactly what we're seeing in the human.
Tell me a little bit about what did you call it?
And
Amplitude bias?
so when you're
Yeah.
when you're doing PCR, you're making copies of the DNA, right? Like photocopies, if you will. And as you photocopy or as you copy the DNA by PCR, the problem is if you have genes that have a very low copy number, you're going to miss those. And so that's how you get amplification bias. With nanostring, it's probe based. So they have a little nucleic acid probe that has 7 different fluorophores on there that binds directly to the RNA. And so because it's probe based and it's fluorescent and it's got 7 different probes, they can ID up to 800 biomarkers at a time in the same sample. So it really allows you to, and the robot does, there's very little, there's like 2 pipetting steps, and you put in the robot does all the washing of the plate, and then the other machine does all the reading, almost like a confocal microscope. So it gets rid of the amplification bias. It reduces the PCR error, the technical errors with pipetting. Those three. I don't know if you've ever pipetted into a 384 well plate with and having to put in multiple things. The chances that you're going to screw up are so high, it's terrible. The cool thing with nanostring now is when I purify RNA, I could send it to 5 labs around the world, and they get the exact same data that I would get in my lab.
So it's less lossy, basically,
Yeah.
if we to use a statistic. Okay, that's very interesting. You talked a little bit about the p values that you get in the lab. For the work we do it on the clinical side primarily, or we model systems, etc. In your lab, when you look at p values, how do you arrive at those, and for what do you arrive at those for?
So we do the basic p-values with a t-test, and then we adjust them based on the kind of the linearity of the data. And we look at a lot of housekeeping genes. So we can we can kind of do an adjusted p-value so that it's to increase the strength of the probability that we're really looking at something that's real. And the cool thing with nanostring, as I say, when you get your data, sometimes p-values are 10 to the minus 6. And the cool thing is we've taken, we do a lot of work in traumatic brain injury, and we've done samples from different cell types, and we've repeated samples, and we've done different doses of drugs, and we can see profound differences in even, identifying the right dose for the patient before it goes into clinical trials as well, which I think is really exciting.
That's fascinating. And just for the audience, I know we're getting deep into the technical here with Mitch. So, a t test for the non stat or non quants here is a test where you compare the means of two groups that have a continuous outcome. And what you're trying to assess is are they equal or not? Right? That's what a t test does.
Yeah.
It was ironically, the t-test was for those people who like alcohol, was created by a brewer at Guinness. And the reason it's called the student t-test is because he couldn't put his name on the papers, so he just called himself student t. It's just a little bit of fascinating history around the
Fun.
t-test. But
Cool.
that's fascinating. That is really fascinating. So because it's interesting, the same methods that I would use being used at a biological level. What other kind of tests, statistical tests, do you do in your lab? You do the t-test. What other kind of statistical
We
work do you
do
do?
a lot of multivariate analysis of proteomics. So when we do mass spec, we look at two things. One is we can do basic t-tests, but sometimes you want to understand how the genes relate to one another. And so we use Jump SES, and we can go in and take the whole data set and try to understand what represents healing and what represents a wound that's not going to heal and which genes cluster together. And it's so funny. I used to I when you have 2,800 proteins in proteomics and you try and do it on your MacBook, the my original MacBook, it said that it was going to take 7 days to do the analysis.
And that's not Monte Carlo. That's just
Uh, and so what I realized was I could do if I did 400, if I got the most expensive MacBook I could find is the one I have now, and I did blocks of proteins like 400, 600 at a time, I could do it in half hour, what have you.
Yep.
The first time I said it with my old MacBook, and I realized it was going to run for 7 days, I was like, oh man.
You got to use the GPUs, Mitch. You got to
Yeah,
get down
yeah.
and use the GPUs.
Well, it's funny because when I at Alira, I had the most expensive computer in the whole company. It was like, and in at MIT, I was used to using supercomputers for, you know, for protein folding and protein dynamics, and then I've always been at the edge of trying to get the fastest computer I could do to do all the math. It's really been fun, but so I find a happy medium where if I take 600 genes or 600 proteins, I can do that with our mass spec. And we've found a whole bunch of genes that seem to say your wound's not going to heal, and others that say you're on the path to healing, which is really exciting. Um, for NanoString, we use a very powerful software package called Rosalind, which allows everything's done in the cloud, and so literally I can go from purifying the art. The kids in my lab purify RNA in a couple of days. NanoString takes less than a day to wash the plates, do all the reading, and then if the supercomputer is not busy, we can have data by the afternoon, by the next day, that's publication quality data. It's just unbelievable. So it's really cool.
No, that's great. It's I, as you probably know, I'm a Bayesian, and so one of the bottlenecks in Bayesian statistics is MCMC. It's computationally intensive. I like
Yeah.
you. I fit one model a couple of weeks back, and it came back and said it's going to take 3 days. And I was like, "This, I'm not sitting here. Like, this is crazy talk." So
Well, the problem is
I wrote
my
some.
was going to go to sleep before it even started to do quick computations.
Exactly, it's absurd. But there's some hacks now. I mean, the cloud-based GPU computing is amazing. And then
Yeah.
in Bayesian circles now, they're taking advantage of some of the new technologies like SIMD and writing new algorithms that approximate MCMC so
Oh, cool.
close that it's you can. I tested one and it cut my processing time down from 3 hours to 7 minutes.
Yeah.
You know, and I, like you, have a powerful computer. I have a M1 Ultra, which is ridiculous, but it's just fascinating, kind of the interplay between computational biologies, what I'm guessing you're kind of doing, and other, how that plays into the framework of machine learning, AI, Bayesian, etc. And we're all kind of benefiting from the technologies that are coming about. Though those GPUs still cost about $3,000, so hopefully they'll get cheaper. So you've talked about sort of the predictive side of things, where you test something and then you can say, okay, our prediction is that this is going to lead to healing on wounds. What I'm really interested in finding out about as well is this wound repair and regeneration part of your background. So an interesting question that we've seen at OWR and we're thinking about is, once the wound heals, how can we gauge the tissue quality of
Yeah,
that tissue, especially in pressure ulcers, right?
yeah.
And how do we build models to kind of say, okay, this person has good tissue, ergo the probability of recidivism is less. How do you kind of think about the biology of that, and what kind of drives that?
There's kind of basic parameters that I think Chendon and others have kind of looked at that suggest that you're really healing the wound and not just covering it. So moisture vapor transmission rate is probably a good indicator of whether you're truly healing the tissue. You're getting a tight interaction between the epidermis and the dermis, and you get good tight junctions, and you've truly, you know, reorganized and healed the wound. The other, what we see, what we can't do in the human case, but in these animal models, we can look at the strength of the tissue after it's healed, right? There's some other markers that are really interesting that I find very exciting that we should think about more. And this is in the skin barrier function space. This is thought of all the time. They think of lipidomics and they do lipid profiling to understand whether you have a good barrier for whether it's better cosmetics to prevent scarring or just have less uh, dermatitis or anything like that. But we should think about the skin barrier function in a healed wound to better understand whether it's truly healed and whether it's going to prevent recurrence. And so I think there's a number of biomarkers, and I think we have some that we haven't submitted patents on that we're submitting that will give you an indication of, do you have the right protein composition to prevent recurrence as well. But as I say, I think the lipid component is really a really important surrogate for trying to understand what's going on there. And with simple, I don't know if you've ever seen they can do like a, a little skin test where they just take scotch tape and they pull off a little bit of the layer of skin, and that's enough to do mass spec on the lipids and to better understand whether you're going to have a good skin barrier function.
Oh, that's
So
brilliant.
yeah,
Let me ask you, what kind of measure would you have? And this is something that I was talking with Harm Smith, who's also a biologist out
yeah,
of the
yeah.
Netherlands. What's the measure for strength of tissue? Like, as a biologist, what measure would you use?
Oh well, it's force in newtons, right? So when you have a unwounded tissue, you're probably getting 70 to 100 newtons, which is I can't convert that to pounds per square inch, but you probably got 70 to 100 newtons, and it's a well intact tissue. When you have poor barrier function, but you covered, it may break apart in 20, 30 newtons. So you can tell that the wound is closed. It looks like there's an epithelial layer, um, decent amount of keratin. It does look like it's starting to remodel, but it really doesn't have the strength to prevent it from opening up again.
That's interesting. So, if I'm hearing you correctly, it's the ability to withstand force. Is
Yeah,
that the
I mean,
measure?
we literally can put them in a biomechanical machine, pull them apart, and measure the force on break. And we can
That's
also look at the
fascinating.
elasticity of the tissue as well.
Interesting, interesting. And kind of jumping from wound repair and regeneration to kind of diagnostics, because this kind of flows in there. Like, what's the pathway to get something in clinicians' hands where they could literally go to a wound that's healed, use a pro, whatever it may be. I don't know. I barely know what I'm talking about most of the time. And in this case,
I,
I'm pretty
I,
sure I don't know what I'm talking about. But like, if they had a probe or something that they could literally test these different parameters.
I think there's a big problem in diagnostics, and I faced it when I started back in 2000 developing wound care diagnostics, which is until the FDA accepts some of these new diagnostics as a primary endpoint, like let's say infection would be, or debridement. Um, if you work on debridement, there's no reason why you should have to go to full healing to know whether you debride a wound, you know. But the FDA forces you to do that, and it's then you end up running these really expensive trials because you have to go all the way to healing. But if once the and I think the wound care collaborative community, I think Vicky Driver has been spearheading this for a number of years, trying to get the FDA to accept new primary endpoints, and I think they're listening. And I don't think they're quite there. I still think they call them secondary, good secondary endpoints. But once we have primary endpoints like wound bed prep, debridement, infection, things that, the problem is you can develop the best diagnostic possible, but if it doesn't change clinical practice and people aren't going to reimburse, if there's no pricing and reimbursement for it, it doesn't matter. So I always, as a scientist, an entrepreneur, when I started in this field 20 years ago, I thought the panacea was getting FDA approval. That's actually trivial. The panacea is actually getting pricing and reimbursement and getting adoption and changing clinical practice so that you really have a foothold. There have been some great analytical tools for scientists, including the moleculite. The moleculite's not perfect. It can't detect all bacteria. It doesn't detect Enterococcus. But the two primary bacteria in a wound are Staph aureus and Pseudomonas. Staph aureus will glow bright red, almost like a ring of fire, with the moleculite, and Pseudomonas is more of a kind of a cyan green kind of color. But it really tells you about debridement. Did you really debride? And so you can go in, light up the wound, and you can sharp debride until you get to fresh granulation tissue and you don't see any fluorescence. And I think that has the chance of changing, you know, providing better antimicrobial stewardship, which I think is really, really important. Now, that said, um, some we see this all the time. Some devices are never going to be, even though they may be accepted in US and there's pricing and reimbursement for it, it's hard to bring some of these cool diagnostic tools to Europe because the payment structure is so different, right? And so the challenge, as I say, it all comes down to how do you change clinical practice and how do you heal a wound faster and how do you get paid for it at the end of the day? And the problem that I face is I love developing cool technologies, but most people will never pay for half the stuff I could invent. And I've had so many great scientific discoveries that would never make it to market because it didn't, the great example was the frowning face on your hot dog package when you had listeria. Nobody wanted nobody wanted it, right? Um, so I think that's the real challenge in working with smart people that understand pricing and reimbursement and how to change clinical practice so that it really benefits the patient. The other thing is that I think a lot of these companies go into wound care when they should have instead of going into chronic wounds, they probably should have gone into surgical wounds because the pricing reimbursement structure is so much better, you know? And so we've seen a number of these companies have really good products, but they've really faced challenges getting enough payment for their product in the chronic wound space. Whereas if they went to surgical wounds or dehiscent wounds, that may have been a much better opportunity for them first, and then try and do DFUs and VLUs and pressure ulcers.
No, absolutely. As a marketing guy, we always, talk about product market mismatch, and you see that a lot of times. You see where, especially, I've seen this with companies that come out of Europe, right? They have a vision of this market that they think is there in the U.S., and they totally misalign their product. And to your point, they kind of get stuck in that channel if they come in initially on that. If I may take maybe 1.5 steps back, you talked about the WCCC and the endpoint of complete wound healing. And I'm going to rant here for maybe 15 seconds because it drives me a little bit crazy as a stat guy. One of my pet peeves is discretization of continuous data or discretization of things like healing. right? It imagines a world where there is healed and then not healed. But that's not how it really works in the real world, right? A door is open or closed, agreed, but it's also a jar. And you can measure how ajar it is. And a door that is 99% closed is very different than a door that is 1% closed, right? And I think a lot of what harms, at least on the statistics side, but also advances in science generally, is this discretization and creating this monochronic version of reality that literally doesn't make sense. You know,
Right.
I think initially I worked in post acute on the marketing side and worked with clinicians, and clinicians do think about wound closure. I mean, they talk about this every day. And to say that that does not matter as a clinical endpoint, when the people actually providing the care talk a lot of the time about it, is just kind of crazy to me. And I think that it will, it could lead to a situation where great products are left out there because, and I'll give you an example. I ran an analysis. There were two products that had the same wound closure rate or the same inferred wound closure rate. But and these were on chronic wounds that had been around at least a year. They had the same wound closure rate or within like 5% or something like that.
Yeah.
But the one product closed those wounds by 85%, whereas the other product did nothing or the wounds got worse. And the notion that those are two equivalent things is preposterous to me. That doesn't
Well,
make any sense.
I think the FDA is starting to, and even many of the clinicians understand that it's great if you can follow these patients beyond wound closure, because at the end of the day, is did it improve their quality of life? Did it prevent recurrence? And I know Dr. Armstrong likes to talk about wound recurrence almost like cancer. It's going to happen. But the point is that if we get to the etiology and we change the pathology of why wounds aren't healing, we probably can prevent recurrence. And great examples of that today are look at all the great um, advances that have been made in topical oxygen therapy. And we're seeing that with devices like that are easy to use, that the patient doesn't mind, having that therapy, you can get better outcomes after, and it's more than just healing. And you're right. It's very hard when I talk to biz dev people at these large companies because they say, "Oh, that's the same rate of healing. We've already got that covered." And I said, "No, you don't." And they don't really understand that. But the same is, it's even worse though when you think about developing a diagnostic. You think that the gold standard should be, sensitivity, specificity, area under the curve, no false positives, no false negatives, and that should be it. But no, as for my first diagnostic, they forced us into saying, we got to show that this will, if this goes off, it'll prevent healing, which is ridiculous. When you're just trying to determine whether there's an infection brewing or not, so that you can treat it correctly the first time. And the problem, as you know, in with a chronic wound infection, which is a deep tissue biofilm infection, except for 50% of the time, clinicians on visual signs get it wrong get 50%. You could flip a coin, you'd have better luck at guessing whether it's infected or not. Whereas we could tell them the early warning signs that there's too much bacterial protease, it's going to liquefy the wound, it's not going to heal. And but that's the challenge with some of these cool diagnostics that are coming out that have all these cool near IR imaging and what have you. Um, it's a lot of these products are getting cleared now. The challenge is pricing and reimbursement, and how do you change clinical practice, and how do you make it affordable so that in Europe a lot of the care is given by nurses. That how do they pay for the device?
That's definitely a problem. I'm originally from South Africa, and I work with wound care, great wound care people down there, And these technologies, they go, we can't get our hands on them. There's no way we're going to get paid. The acquisition cost is crazy, and our currency is weak. And so it creates a barrier of entry to these products. And what we've tried to do is to talk to companies and say, listen, here's a great site. You're going to get a lot of data out of the site, and it's going to give you insight into a population that maybe you did not consider, you know. And so I just think there is definitely these borders where reimbursement is just an issue. Even in Europe, I lived in Germany, I lived in the United Kingdom, and it's two insanely different systems
Right.
of care. The other thing I'd like to talk about, so we've talked about predicting which wounds would heal or not. We've talked about quality of tissue. Walk me through kind of debridement. I mean, the way I came from, again, the sort of, I'm not a clinician, but I worked with clinicians. When I think of debridement, I think of two things, right? I think of the Santils of the world, the enzymatic debridement. I think of the mechanical debridement. As a scientist, and I've heard this story many different times, I like to ask different people, how does debridement work to get better outcomes in wound care? What is your sort of theory of that?
Yeah, I think when you have the very hard eschar, it's it can be very deleterious to closing to providing proper wound healing because you don't have good oxygenation. You have all this dead, devitalized tissue that's it's kind of in the way of trying to close the wound. We get um, uh, Escar from Dr. Serena all the time, and we get the green slough stuff, which is a different story, although we'll get to in a moment. Then we get this rock hard black Escar that is just impossible. It's like a rock. And then when you have the green slough, the problem is you have all these degraded enzymes, you have a lot of proteases, and it's going to cause damage to the tissue so that it can't properly heal. It's going to delay healing in a slightly different way. And so the way that I think about this is enzymatic approaches can be really good. The problem is with many of them, the ones that really work can be quite painful, right? And so it has to be done either in the OR or you have to be under anesthetic or what have you. Santil takes forever to work. It's like watching grass grow. It takes like 30 days, up to 30 days to bride a wound. It's not very practical, but it was a great moneymaker for Smith and Nephew and before that Health Point. Some of the new approaches are scalpels have been around for a long time, but it's usually used in the OR. It's not great for clinics and other things like that. There are other kind of in between methods like the LNR little mitt that and these kind of gentle ways of micronizing the tissue so you can get rid of that material. And then it goes back to the old, the I always think that Barry Wolfenson, when he was at Derma Sciences, it was just a genius in marketing. And he I don't know who in his group came up with autolytic debridement, but it was showing that even honey can help debride a wound by improving the autolytic nature of the wound. The other thing that honey probably was doing was preventing the proteins from unfolding and causing eschar in the first place, right? But it wasn't a good marketing story, so you don't talk about that. But there's a lot of new products coming out on the market that prevent eschar from forming in the first place that I think is a really neat advance. And as I say, it's kind of a balance of homeostasis. I think that's one of Harm's favorites, pet peeves. He always talks about homeostasis. But I do think that if you can put if you can control the host protease and bacterial protease in the inflammatory phase, you're probably going to prevent a lot of that eschar formation and require less debridement. So that's one. And then, as I say, I hate to say the old adage of keeping a moist wound environment is probably still very real. And so making sure that you don't have devitalized tissue forming is going to allow for cell proliferation and the progression towards healing as well. So I think there's the smart people are starting to think about how to prevent it from happening altogether. And the others are saying that enzymatic debriding opportunity is a large market like MediWound and what have you. We can do, if you can do it faster, if it's safer, if it's more gentle, if it doesn't prevent eschar from forming in the first place, you're really gonna, it's gonna pay dividends.
Absolutely, that's fascinating. I think what I find fascinating about how biologists like you and Harm look at this is that you guys get into sort of the micro of what's going on, and it's just fascinating to me the different ways that you guys are able to tell the story of what's going on. And hopefully that's that the audience can drill into from this podcast. The final area that I think is fascinating and came up on your resume was the idea of tissue infection. So, for example, in post-acute world, one I would say number one reason that patients go back to the is because of infections in pressure ulcers predominantly. If you could talk a little bit about tissue infection in the context of, let's say, pressure ulcers, what's the process that goes on there, and what kind of technologies are coming down the road that could help with that?
So the pressure ulcers are such a difficult area to study, because as soon as you start giving the patients better care, you know, they're going to have less pressure ulcers. So the prevention is probably the most important thing for pressure ulcers to begin with. And then, having these techniques to look at good skin barrier function from the start. If you have good skin barrier function, you're not going to get an infection. And so maybe that comes, once again, it comes down to prevalence, it comes down to prevention. And but once your wound's infected, if it's a deep tissue infection, your body's just not going to be able to fight that off, and you're going to have to debride it. You're going to have to treat it somehow. And the question that I always look towards is, people love, clinicians will give you oral antibiotics, but oftentimes they still need an antimicrobial, because, and what we're finding about in this antimicrobial space is, the problem with antimicrobials is that they were all developed mostly using planktonic bacteria. And when you dig deeper into whether any of these products really work in a biofilm model, especially in vivo, we 3D print biofilms and put them in a diabetic mouse, and they're really hard to kill. And hypochlorous acid, which is sold a lot in the US under a number of brands, Vosh in particular, it's great at killing planktonic bacteria, but it's not great at killing biofilm. And there's new generations of hypochlorous acid, new generations of products that have focused solely now on trying to treat the biofilm and not just these free floating bacteria. So we have a fighting chance of really getting in there and being able to clean out the wound, which I think is great. And there's a whole bunch of different ways of going about it, whether it's an acid solution from Devex or something else. But there's a number of ways that you can go in, eradicate the issue so that it can go back to healing. Otherwise, it's just going to fester.
Absolutely, that's fascinating. I think what really could bend the cost curve, and at OWR we think a lot about costs, right? What are the costs to the system of
Right.
these ulcerations, these wounds? And we do a lot of work. We even had a poster back in the day on a probabilistic model for estimating wound care cost over time that almost no one was interested in, but we were. I think it's really hard to go into a population, risk stratify, and identify exactly who's going to get wounds, right? It's so heterogeneous, right? And I think a lot of people are looking at that as a panacea, and I'm thinking to myself, that's an almost impossible fight. However,
Well, I do think there's a number of diagnostics that are coming out that are going to tell you whether you're apt to get a pressure ulcer. And there's some great science at UMass Med. They have a number of kind of markers uh, that, with an algorithm, suggests that they can predict whether you're going to have a pressure ulcer or not, right? And there's
interesting.
other methods to do it as well. But I think that's where preventing the skin breakdown, and there's a number of new kind of rocker technologies coming out, so that there are beds that are going to have kind of a wave action, so that you're never continuously,
Gotcha.
when of all the leaf technologies, things like that, that just
Hmm.
say that the patient's not moving, which is, it's it it's part of the solution, but it just makes it harder on the caregivers, right, to monitor that
Hmm.
and try. focus their protocol on turning the patients. But I think more importantly, it'd be great if they could predict when these things are going to occur so they don't occur in the first place.
No, it would be, and I think where I come from is, and maybe this is my bias. A lot of the data we look at is based on data for, let's say, Braden scores, right? And we have each nursing home has to have a Braden score at X time point, and so what we do is we stack up those Braden scores, and then we infer risk from the Braden scores because you can have two people who score 7 or 10, but the way they score it is different, right?
Right.
And so you're trying to estimate the latent variable of the repeated measures, right? And so maybe I'm being unfair because what you're talking about is biomarkers, right? Talking
Right.
about something that's fundamentally different. Maybe, the one that comes to mind is SEM, for example, right? And I think, well, fair enough. I think, if that technology can come along and we can prevent a lot of these wounds, I think that's going to be huge. I also think what's also going to be huge is being able to stop these wounds from reopening. If you look at the data, and we've worked with payers, etc., you see a wound happen for the first time, and what really piles up cost are the constant reopenings, right? And they just can't stop these wounds from reopening. And so, listen, the golden standard is to stop it from happening in the first place. I think just as important is how do we kind of bend that recidivism rate downwards? What are the predictors? How can we identify, not only at the patient level, but at the tissue level, the people that are good healers versus bad healers?
Right. No, that
Yeah.
makes sense.
Yeah, absolutely. So we've got about 4 minutes left here. My goodness, the time has flown. It's crazy. I have one question for you, and I might sneak in another question for you. What are you going to be working on for the next year and a bit? And if you had infinite time and resources, what is the one thing that you would attempt to solve for in wound care?
I think we're really focused this year on translating what we've learned in the diabetic mouse and starting to do testing of patients in these pilot clinical trials. So we can get a small biopsy before the wound, when we realize that you have the run-in phase and the wound's not healing, get a little bit of tissue, um, do the treatment, give it a couple weeks. Once you see a response, take another biopsy and actually understand the mechanism of how your product's better. And the reason why I think this is really important in the camp space is there's a lot of placental derived tissues out there. There's 200 plus placental derived tissues. And my belief is that, a clinician may say they're all the same. They're not. The minimal processing really does matter. And so product X versus product Y are hugely different in how they're going to affect wound healing and the rates and the final integrity of the wound at the end. And so being able to tell a story of what the mechanism of action of how your product works will also hopefully help you get reimbursement as well, so that the payers will pay. There's this huge other issue of the the in this space where there are some bad players that have really overcharged for or putting these products on wounds that aren't going to benefit from them. But these products really do work if you put them in the right wound at the right time.
Mm-hmm.
But what I can say is there's, I don't know if there's 200 companies, but there's at least 200 products out there, and they all are actually quite different in how well they work. And so that's, I think that's it's a little bit because there's this mystique on how these products are minimally processed is really art. It's an art form, and some people do it well and others don't.
Absolutely. Yeah, that makes sense. I think, again, homogenizing or trying to, I think one of the issues is reimbursement again. It goes back to reimbursement. In order
Yeah.
to pay for it, we got to put them in a bucket.
Yeah, everything has to be in a bucket. Yeah, it's neat and tidy. That's the problem with the 510K process, is that you get clearance on a product from a product that was pretty good, but your product may not be as maybe inferior.
Hmm. Hmm. No, or it might be extraordinary at one thing, but not as good at another thing. So it might, cause the wound to close, to go towards closure or approach closure quickly and efficiently and cheaply, but it might not help a subset of those wounds to totally close. And then you have to bring something else that has that ability to close wounds.
The
Um,
other, the other thing that we're doing is we're starting to bundle products in our trials to make it more cost effective to get these products to market. So, and many people realize that your product may be good for one phase of the healing cascade, but not the other part. So, having multiple products in the trial gives you the kind of continuum that Integra was hoping for with a lot of their products that they put together, where you do wound bed prep, you do call the inflammatory phase, and then you go on and do proliferation and then angiogenesis or what have you, tissue remodeling. So I think that's where we could really benefit from people starting to design trials with multiple indications. The other thing is the that great defender boot that's great for patients with diabetic foot ulcers. When you put them in the boot, they are getting a better standard of care,
mhm.
and they also make a lot of these cell tissue products do better in the healing cascade because you're getting better offloading because it's better compliance. So that's where these bundling of these great technologies is really going to pay dividends for the patients.
No, that's brilliant. Last question. If you had infinite time, infinite money, you're a man of many interests, scientific and otherwise, what is the one problem that you would attempt to solve for?
Oh my gosh. Um, there's so many. I think one thing that I've learned in traumatic brain injury is studying chronic inflammatory disease in a wound will help you understand traumatic brain injury. And there's a lot of parallels between chronic inflammatory disease in a wound and all these other chronic disorders. And if we can bridge the gap in our understanding down to a pathway, we may be able to help other patients with other chronic inflammatory diseases. So that would be really cool.
Oh, that's a great answer. That is great. I like that. Well, Mitch, it's been an absolute pleasure. This hour has flown by. I appreciate all the knowledge that you dropped in 60 minutes. I think our audience is going to get a lot of value out of this. And to be honest, I'm thinking of bringing you back on because I just feel like we barely touched the surface of a lot of things that you're doing. And one of the things we're going to be doing is having two people on at once who,
Cool.
I would love to have you and Ham kind of just drill down and it'd be
I, I,
great.
I just talked to him 2 weeks ago. But Zweli, I think what you do is amazing, and thank you for having me on. It was a real pleasure.
All right, thank you so much, ladies and gentlemen. That is the great Mitch Saunders. I enjoyed it. Hopefully, you enjoyed it, and you can check him out on LinkedIn. He's posting constantly about science. He even writes books. People, this is a man of many talents. So, thank you again, Mitch,
Take
and
care.
thanks to the audience for your time. Cheers.
Honored. Cheers.